Hello everyone, my name is Prajakta Dhage. I am from Department of Zoology, KTHM College, Nashik. Today I'll be taking a practical for class T.Y.B.Sc semester 4. The subject is practical paper 3. The module that we are going to deal with is general embryology, and the name of the practical is studying the types of blastulae and gastrulae. This we'll be studying with respect to Amphioxus, frog and chick. Let's first begin with seeing what is a blastula or what is the process of blastulation. So, after a ball or morula is formed, post formation of morula, the process of blastulation takes place. Now, process of blastulation means the formation of blastula. Blastula is a hollow sphere of cells, which these cells are also referred to as blastomeres. This is produced during the development of an embryo by repeated cleavage of fertilized eggs. The cells of blastula form a layer of cells called as blastoderm. This blastoderm encloses a fluid-filled cavity which is referred to as a blastocele. Now let's first begin with the study of the Amphioxus blastula. Let's see what are the characters of that. So, if we talk about the egg of Amphioxus, the egg is oligolecithal egg which contains little yolk, and the type of cleavage that is seen is holoblastic cleavage. An understanding of blastulation in Amphioxus demonstrates the fundamental developmental process in the simplest forms, since the egg contains the little yolk. As a result of cleavage, the blastomeres, which means the cells, are formed or that are formed are not of equal size. The blastomeres near the vegetal pole of Amphioxus blastula are slightly larger than those of the animal pole. After the formation of the blastula, the cells arrange themselves into a simple cylindrical epithelium enclosing the blastocele. You can see the blastocele over here, large blastocele over here, single layer of cells that is surrounding the entire blastocele. Okay. Consequently, the cavity is centrally located within the ball of cell. The cells from the vegetal pole of the egg will become the floor of the blastula, or will also be referred to as hypoblast cells. And these cells are going to form the future endoderm of the embryo. These cells from the region the cells from the region of animal pole will form the roof of the hypoblast. Okay, so these cells I'm talking about. Now these cells are going to become the epiblast layer. So this particular layer is going to be the hypoblast layer, and this one is going to be the epiblast layer. Okay, so the epiblast is going to give rise to epidermis, the neural cells, the notochordal cells, and the mesodermal cells. So basically all the origins of the epiblast are going to be either mesodermal or the ectodermal. Okay, so this is about the blastula of Amphioxus. Now let's move ahead with the blastula of amphibians, and the example that we are going to deal with is frog. Okay, so in frog there is a large amount of yolk present. So what happens, the blastomeres at the end of the cleavage show clear-cut difference in size. If you see the blastomeres or the cells over here are smaller as compared to the cells over here. Why is it so? Because of the large amount of yolk. This egg is a telolecithal egg also. So the cleavage furrow is going to pass slower, and this is going to result into the formation of two different sizes, or the size of the cells is going to be different. Okay, the cells are also arranged in a layer surrounding the cavity in the interior, but the layer is of very uneven thickness. The cells formed from the vegetal pole of the egg are referred to as hypoblast and do not have pigmentation and are much larger than those at the animal pole. They push the blastocele into the causing this cavity to become eccentrically located. So basically what happens over here is, because the size of these megameres or the cells which are present at the vegetal pole region is larger, this particular blastocele is pushed towards the animal pole region. Okay, so and now as it is not centrally located, this particular blastocele is referred to as eccentrically located blastocele. Okay, so the large yolk-filled cells are referred to as megameres, and the smaller cells are referred to as micromeres. Okay, and micromeres are going to be present at the animal pole region, and they are going to form the epiblast. In frog, the darkly pigmented cells derived from the animal pole of the egg give rise to the future ectoderm of the animal. Further division of the ectodermal regions provides cells for the epidermis and the nervous system of the embryo. Here, in such cases, the blastoderm is no longer a simple cylindrical epithelium made up of 2 or more cells thick. In the interior, the cells are loosely connected to one another, while they adhere to one another very firmly and very tightly towards the external surface. The adhesion of the cell is due to presence of the cementing material. Here you are going to have, on the outer side it will be tightly bound, on the inner side the cells are going to be loosely bound. So this was about the blastula of frog. Let's move towards the blastula or the blastulation in case of chick. Okay, here as the egg is megalecythal and telolecythal and heavily laden with yolk, what happens is during the formation of blastoderm, cells are incorporated into the superficial layer, the epiblast. A small number of blastomeres remain underneath the layer and the yolk is formed by the adhesion of the cell. This layer is called as hypoblast, separated from the epiblast by a cavity. This cavity is in birds and reptiles corresponds to the blastocele of the amphibians and fishes. In birds and reptiles, this cavity does not appear under the central part of it. No cavity is formed under the layer of blastoderm in the periphery of blastodisc. The blastodisc in reptiles and birds is subdivided into 2 parts. The central part called as area pellucida and the peripheral part referred to as area opaca. So this particular portion is area pellucida and this particular portion is area opaca. The names indicate that the central part appears more transparent in the living embryo. This part is going to appear transparent, owing to the process or presence of the cavity under the layer of blastomeres. The peripheral area is opaque because the blastomeres rest directly on the yolk. So this was about blastulation in the chick embryo. So see, gastrulation. Gastrulation is the phase. So gastrulation is a phase that occurs in early development during which the blastula, which generally has epiblast and hypoblast, and a hollow sphere called as blastocele, is reorganized into a multi-layered structure known as gastrula. So basically, when we talk about gastrulation, there is formation of 3 germ layers, which is ectoderm, endoderm and mesoderm. Also, there is a structure that is going to form, or the cavity called as gastrocele will be formed. So let's begin with the structure of Amphioxus gastrula. So let's begin with the gastrulation in Amphioxus. In Amphioxus egg, the segmentation results in the formation of centrally placed blastocele. We have already seen this. The beginning of gastrulation is indicated by flattening of the vegetal hemisphere. The flat area then invaginates and forms archenteron, having its opening to the exterior in the form of blastopore. So this particular structure that you see over here is referred to as blastopore, and this is the gastrocele or archenteron. Now the blastopore is bounded by dorsal, ventral and lateral lips. So these structures that you see over here, these are nothing but the lips of the blastopore. Archenteron increases gradually and ultimately occupies the entire space of the blastocele. The embryo at this stage is referred to as the gastrula. So here what you see is the gastrula of Amphioxus. Okay, this is the gastrula of Amphioxus. Now let's move towards the gastrula of frog. Now, as we have already discussed, the egg of frog is mesolecithal and telolecithal. So here the cleavage results into 2 kinds of blastomeres, which is megameres on the animal pole and micromeres on the animal pole and megameres on the vegetal pole. So the micromeres divide rapidly and flow down over the ventral surface of the blastula. This process is referred to as the epiboly. During this process, megameres are covered by the micromeres, except at a place called as the yolk plug. So if you see this megameres, all they are covered by the micromeres. Why? Because these micromeres are moving over the megameres. Now, somewhere near the yolk plug, the invagination starts the formation of archenteron. And as the invagination proceeds, the original blastocele is nearly removed or is nearly obliterated or gone because of the inpushing of the layer of cells. Okay, so this was about the gastrulation in the frog. Let's see the gastrulation in case of chick embryo. Now, we know that the eggs in chick are megalecithal and telolecithal. So what happens is entire development or the early development is going to occur in a small disc, okay, which is in a small disc. So the cleavage occurs in the germinal disc or blastodisc, which is differentiated into area pellucida and area opaca. Now, what happens is due to proliferation, delamination and convergence, there is formation of primitive streak. Because of these 3 processes or the cellular movements or increase in the number of cells, there occurs the formation of primitive streak. So this basically what you see is entirely a primitive streak. Now primitive streak is characterized by presence of primitive groove. You can see a groove running over here, white color. This is nothing but a primitive groove, and on both the sides it has primitive folds. Then often primitive pit appears in the form of depression, and the raised area around the primitive pit is the Henson's node. So towards the anterior region you have this particular structure referred to as the Henson's node. From primitive streak, the mesoderm grows out laterally and anteriorly by the process of divergence. So basically this is how the chick embryo or gastrula of chick embryo is going to look like, and it is going to be at 18 hours of development, where you can see the formation of primitive streak. So the basically the cells are moving from backward towards the anterior side. So this was all from my side about the practical of blastula and gastrula of Amphioxus, frog and chick, and thank you so much.